Good morning, my dear students. How are you? Are you now ready to learn new things? I hope it's a yes. Today, we will be tackling about biotechnology and genetic engineering. But before we proceed for today's discussion, let's have a short review of what we have discussed last week. Can you still remember what was our discussion last week? Very good. Last week, we discussed about mutation. Mutation refers to a change in the molecular structure of a gene or the physical structure of a chromosome. There are two types of mutations that can occur in gamete cells. First is the gene mutation, which is a permanent change in the DNA sequence that makes up a gene. Second one is the chromosomal mutation, which occurs at the chromosome level, resulting in gene deletion, duplication, or rearrangement that may occur during the cell cycle and meiosis. It may be caused by parts of chromosomes breaking off or rejoining incorrectly. Are all mutations harmful? What do you think? Not all mutations are harmful. For instance, mutations have been responsible for antibiotic resistance in bacteria, sickle cell resistance to malaria, and immunity to HIV, among others. Mutations also cause diversity. Things like hair color, baldness, freckles, and beauty spots are all brought about by mutations. So what does GMO mean? Right. GMO stands for genetically modified organism. Have you eaten a genetically modified food today? It's probably a yes. Here are some examples of genetically modified organisms. The glowfish was the first genetically modified animal to become available as a pet. The grapefruit is a relatively new fruit, which is a genetic cross between an apple and a grape. And the last photo is probably the most remarkable example of how far science is able to go with modern DNA and cross-fertilization techniques. The lion is a cross between a lion and a dog. So, how are GMOs created? Let's watch this video.
So you want to know how a GMO is made? You're not alone. It's one of the top questions about biotechnology. There are different ways plants can be genetically engineered. We'll use the story of the papaya in Hawaii to walk you through one of them. Step 1. Determine whether genetic engineering is the most effective way to solve a plant's problem, such as a disease or damage from pests. For centuries, traditional plant breeding has been used to fight pests, environmental stress, such as drought, as well as to improve taste and nutrition. The reasons to develop GMOs are no different today, but GMOs can be created more quickly and more precisely. Other breeding methods can take decades to produce results and aren't able to be as precise in focusing on one genetic trait or isolating a solution to a specific problem. Since the 1940s, the Hawaiian papaya industry had been battling a problem, the destructive and rapidly spreading papaya ring spot virus, aka PRSV. PRSV deforms the papaya fruit and in young plants destroys the plant's ability to produce fruit at all. PRSV is transmitted by insects that feed on the fruit. Many methods were deployed to try to stop the destructive virus. In the early 1960s, farmers moved papaya production from one Hawaiian island to another, from the island of Oahu to the big island of Hawaii in the Puna district. The virus ultimately reached Puna. Other methods didn't work either, including using insecticides for the insects spreading the virus, giving the plants a mild strain of the virus to create natural inoculation, or destroying infected trees. The virus just continued to spread. 53 million pounds of fresh papaya were being produced annually when PRSV was first discovered in Puna in 1992. By 1998, that amount had been cut in half to 26 million pounds. The crops and livelihood of farmers were being destroyed in front of their eyes. Moreover, for consumers, the availability of fresh papaya and papaya juice for fruit, salads, smoothies, and other products were threatened. A powerful new tool, biotechnology, was now available to fight back. Farmers and researchers turned to genetic engineering beginning in 1985 for help. Step 2. Identify the gene or genetic material that could solve the problem, like providing resistance to the disease or pest, and study the genetic makeup of the plant that needs the trait. Researchers and farmers had to find a way to make the papaya plant resistant to the deadly virus. PRSV resistance was the genetic trait needed to solve the problem. Researchers knew that if plants contained certain genetic material from the virus within their own DNA, they were protected. If they could insert a copy of this gene from the virus itself into the papaya's genetic makeup, the plants would be protected. Step 3. Copy the trait from a donor organism and implant it into the plant's DNA. Researchers isolated a PRSV gene from the virus itself. Then they needed a way to get the genetic material into the papaya plant's own DNA. Researchers deployed a commonly used technique in biotechnology called transformation, a process by which a copy of genetic material from a specific trait is inserted into cells. There are many ways to transform a cell. In this case, the researchers used a high-speed particle bombardment method. Through a high-pressure insertion device, the gene from the virus was inserted into the DNA of the papaya seed. The genetic material made the plant resistant to the ring spot virus, just as a flu shot makes people resistant to the flu. The researchers were able to successfully transform 17 plants. These plants contained this gene from the virus and produced a protein from the virus that helped the plants become resistant to the PRSV infection. Step 4. Plant the new seed and test it. The final step was to plant the seeds and carefully test the resulting plants. One line of plants demonstrated consistent resistance to the virus. After several years of testing, it was determined that this line performed well on the farms, was safe for people to eat, and was of commercial quality. Success was achieved. One variety of transgenic papaya, the rainbow, yielded about 125,000 pounds of marketable fruit per acre per year, 25 times the amount of fruit produced per acre by a non-GM variety infected by PRSV. These tests took about 7 years, including food safety and environmental review by the FDA, USDA, and EPA. Once regulatory approval was gained, the Hawaiian farmers chose to start using the new papaya seeds. The result? The harvesting of the new PRSV-resistant papaya began in 1998, and by 2001, the industry had rebounded, producing 46 million pounds of fresh papaya, free of PRSV. This kept the vibrant papaya industry alive in Hawaii and ensured consumers had access to high-quality papaya and papaya juice at a reasonable cost. Genetic engineering allowed the introduction of PRSV resistance without altering other traits of the papaya or changing the plants in other ways. The plants were exactly the same in nutrition and safety. As of 2010, 70% of Hawaii's papaya industry is GM papaya. GM papaya has been grown and consumed in the U.S. for more than a decade. This is just one GMO story. We hope to share more with you. Please share your comments.
So, what was the video all about? The video showed how biotechnology is applied in curing the papaya ring spot virus or the PRSV. Through genetic engineering, they were able to produce RSV-resistant papayas. So what is biotechnology? Biotechnology refers to any biology-based technology which uses organisms or their parts to make or modify products or improve plants, animals, and microorganisms. It includes the manufacture of more complicated medical products such as hormones, antibiotics, and vaccines. This aspect of biotechnology involves the manipulation of organisms like bacteria, plants and animals, and their products to perform important functions or provide useful products. It involves the technique of manipulating the DNA of an organism. This is called genetic engineering or recombinant DNA technology. This manipulated DNA is introduced into a new DNA of another organism, making it a transgenic organism. You will understand these concepts better as you go along this lesson. These are some creations of plant wizard Luther Burbank. Even if you've never heard of Luther Burbank, you've probably tasted his work the last time you ate a French fry. In the early 20th century, Burbank, who was born on March 7, 1849, created over 800 varieties of fruits, flowers, and vegetables. The plant wizard, as he was called, had a unique approach to horticulture that was part Darwinism, part Thomas Edison. And while his failures often sound like something out of a sci-fi novel, we are still eating many of his creations today. It's activity time! Create your own GMO. In creating your own GMO, you are going to draw a picture of your engineered organism and write a paragraph addressing the questions below. First, if you could engineer an organism, what would you engineer and why? Second, what impact would your genetically modified organism have on the environment or society? You can send your created GMO through my email account probiros.9422@gmail.com. And that's it. Thank you for listening. God bless and enjoy the rest of the day. Goodbye!